Descending route. This refers to invasion of the renal parenchyma through hematogenous seedling of the pathogen. This accounts for 5% of the total UTIs. Although most infections affecting kidney are acquired by ascending route, certain infections occur with in association with pyelonephritis often indicates a descending route. For example, Staphylococcus aureus, Salmonella. Then laboratory diagnosis is the second question. This is the first question, which was pathogenesis. Next question, laboratory diagnosis. See, it is clean voided midstream urine, suprapubic aspiration in catheterized patients. Clean voided midstream urine, it is common specimen for UTI collected after properly cleaning urethral meatus or glands. Suprapubic aspiration from urine from the bladder, it is a most ideal specimen. It is recommended for patients in coma or infants. In catheterized patients, urine should be collected from the catheter tube but not from the urobag. Direct examination, glow wet mount examination, leukocyte esterase test, nitrate reduction test, gram staining. Wet mount examination, it is done to demonstrate the pus cells in urine. Leukocyte esterase test, it is rapid and cheaper method that detects leukocyte esterases. Nitrate reduction test, nitrated using bacteria like E. coli gives a positive result. Gram staining of urine is not a reliable indicator. The bacterial count in urine is usually low, pus cells rapidly deteriorate and they will not be seen well. Culture media, it is only the main points. Culture media, cast concept of significant bacteriuria. Quantitative culture media, colony appearance, where lactose fermenters such as E. coli and Klebsiella, they produce pink colonies, McConkey agar and yellow colonies on clade agar. Identification, colonies grown are identified either by automated identification methods such as MALDI-TOF or Vitek or by conventional biochemical tests. Then, Proteaceae infection. Proteaceae comprises of Proteus, Moragina and Providentia. It consists of naming of H and O antigens. The H antigen is naming of O antigen. Pathogenesis, saprophytes, commensals infections produce cervites and stones in bladder. Cervite stones in bladder. Only this much we have to do for Proteus infections. Laboratory diagnosis is neomorphism, order swarming. Okay. Identification, ICQT test, AST. See, Proteus is a species which shows pleomorphism. Naming of H and O antigens, they are used to denote the flagellar and somatic antigens of an organism respectively. H antigen is named from the ability of the flagella strings of Proteus to grow on agar as a thin film resembling the film of breath on glass. O antigen, the thin film is not absorbed when the strains carrying only the somatic antigen grow on media. Then saprophytes, commensals, saprophytes, nature and they are isolated from decomposing animal matter, CO waste and soil. Commensals, they are frequently present on moist areas of skin, intestine of humans and animals. Infectious product, they are opportunistic pathogens associated with urinary wound and soft tissue infections and septicemia. Cervite stones in bladder, Proteus, enzyme which breaks down urea to ammonia that damages the renal epithelium and makes urine alkaline. Okay. Then laboratory diagnosis. See, it is pleomorphism, which we had already discussed. Order, they produce fish or seminal order. Swarming, Proteus has the ability to swarm on solid media such as blood agar. Identification of Proteus from colonies is made either by MALDI-TOF test or Vitek test. AST, it is performed by either disk diffusion test or by automated MIC method or